Hello. DNA is one of the molecules which is dynamic. It is double-stranded. It can become single-stranded, maybe completely or partially, and then it can reform into a double-stranded DNA. The molecular basis for this is to understand the chemistry and see it in light of charges and groups maybe. Hello, I'm Dr. BCMR, Assistant Professor at Shastra. We'll have a look at the chemical basis or try to understand in very simple terms about how DNA behaves the way it does or why DNA behaves the way it does. First thing we'll just look at it in the form of cartoons just for simplicity so that we will be able to better understand why DNA behaves that way. Here is the depiction of a double-stranded DNA molecule. The blue lines indicate the phosphate backbone. Assume that they are in the anti-parallel because they are complementary double-stranded DNA. And the black ones indicate the hydrogen bonds. It will be easier for us to understand what keeps these two strands together and what is opposing that. The two strands are kept together by cohesive forces, just to imply that these are bound together, are the hydrogen bonds. There are numerous bonds, and that is one of the important consideration we should have. More number of hydrogen bonds will keep because of the sheer number, and AT is with 2 hydrogen bonds and G and C are base paired with 3 hydrogen bonds. So its simple way is if I told you that there is a 100 base pair DNA molecule. DNA, when I say, it would imply double-stranded, unless if we want to denote something as single-stranded DNA, then we should have written as ssDNA. If it is not, it is implied as double-stranded DNA. So if there is a 100 base pair DNA molecule there, and it has about the proportion of adenine in that DNA is about, say for example, 15%, and then I could ask you to calculate the number of hydrogen bonds between these two strands. So these are some things, questions can always be made in such a way. Getting the point is the hydrogen bonds are the ones that are keeping the two strands together. At the same time, there are also kind of repulsive forces, and those are the highly negative charged phosphate. Say on the backbone we have some negative charges, something like this, because of the phosphate group, and you cannot keep those two phosphates two strands too close because there is a repulsion between the negative charges. You can just imagine that way. So the cohesive forces, that is let me finish this, negative charge on phosphate groups. So the cohesive forces outnumber the repulsive forces, therefore the two strands are together, and it all depends on the conditions that the DNA is present in. Let's see how the structure, what are all the factors that play a role in maintenance of the structure or the changes, how the DNA can change its structure based on the conditions. One is inherent within its sequence itself. It could be because of high GC content. By now it is very easy to understand that the more number of hydrogen bonds that are present between the two strands, the more, say for example, this is 100 base pair DNA and this is also 100 base pair DNA. So number of nucleotides is same, but then the composition could be different, because of which the number of hydrogen bonds could vary. Say for example, in the molecule X, and we'll call this molecule Y. In molecule X, the AT is molecule X is rich in AT, and molecule Y is rich in GC. Then obviously we know that adenine makes only 2 hydrogen bonds with thymine, and whereas there are 3 hydrogen bonds between guanine and cytosine. So if we look at in that sense, we know that the molecule Y, which has higher number of GC content, because of which we have 3 hydrogen bonds. Therefore, it is likely that the molecule Y is more stable or remains long together or in double-stranded form in many different conditions compared to that of molecule X. That is how GC content can play a role. Here, when we say about GC content, it is implied in the sequence, right? Because of the base compositions, the stability is changed. We'll also see another molecule in another condition, rather say for example high salt concentration. So here I have 2 DNA molecules, one is X and the other one is also X. They are exactly the same number of the composition is same, the sequence is same, number of hydrogen bonds therefore is also same. What is different, however, is the presence, in which conditions they are present. Assume that the molecule in this condition will say as low NaCl salt concentration, and another one we'll call it as high salt concentration in solution, say. And we remember that we have cohesive forces, that is the hydrogen bonds, and we also have something like repulsive forces, that is because of the negative charges on the phosphate groups. So in which case, now the question is, in which case DNA would remain more double-stranded? About denaturation, about the heat, we will discuss in the next discussion. So here, say NaCl in their ionic form, they can form sodium ions. So the sodium ions can coat the DNA something like this, right? At least in theory, we can it's easy for us to visualize. The more number of sodium ions, the more number of ions coming near to DNA on the phosphate backbone, because the phosphate backbone has a lot of negative charge. And if we assume, remember, or for the sake of discussion, we will put it as the repulsive cohesive forces are, say for example 20 units, and the repulsive forces are some 10 units. Okay, so because the cohesive forces outnumber the repulsive forces, DNA is in double-stranded DNA, in double-stranded form. When we have sodium ions in high in concentration, like in the high salt concentration solution, what we see is more number of sodium ions are bound here, which means the negative charges are actually reduced. That means these might probably could become something like 5 units compared to the other one. What I mean to say is, we have in conditions of high salt concentration, there is reduced repulsion, because of which there is more cohesion between the 2 strands. In the other case, where it is a low salt concentration, the repulsive forces are not so reduced, so they still remain. They are highly probable to become single-stranded DNA upon change in conditions or temperature. So this is a good way of trying to see these things. On the contrary, I missed a slide. Excuse me. So on the contrary to salt concentration, or the effect of salt concentration on the stability of DNA, in salt concentration we are reducing the repulsive forces using the sodium ions, because of which the two strands tend to be together for a longer duration, or even with increased heat. Whereas in alkaline conditions it is different. When you say alkaline conditions, we would mean that the hydrogen ion concentration is low, which would also mean that OH ions are high in number. When we have, say, in conditions, it's the molecule, same X molecule in one of the cases we have the pH is high, or say 7 for example, and in another case we have pH of some 9 or 10. Let's take it as 9. In compared to the pH 7 and pH 9, we can expect that there are more number of OH ions in the solution here. What these OH minus ions could do is to take away the hydrogen ions from the nitrogenous bases, and because of which the nitrogenous bases would be in a different form, because of which they would fail to form hydrogen bonds with other nitrogenous bases. That means we're actually essentially we're disrupting the hydrogen bonds. So we are actually, in that sense, we are reducing the cohesive forces. We are reducing the cohesive forces, and repulsion, assume it is the same, and then we would expect that denaturation would occur here. Say when we do experiments like isolation of DNA, we use alkaline lysis method. Isolation of plasmid from bacterial cells, we do have these principles in there. We are using the alkaline conditions will allow ready denaturation of nucleic acids. That is what we should probably remember. I hope you understood how the GC content can affect the DNA. Increased GC content, we have increased stability of the two strands or double-stranded DNA, and increased salt concentration will also increase its melting temperature. Alkaline conditions will favor the denaturation of double-stranded DNA. Thank you very much.